You are about to watch a very passion-filled conversation between myself and Martina. I envy Martina because, for one, she is actually incredibly intelligent, but she also did physics for her undergraduate studies. I wish I did physics for my undergraduate studies because it's very, very easy to get carried away in biology by the finer details. But Martina, with her physics background, is able to keep a more general perspective on things, a more deeper and fundamental understanding of the way the universe and biology as a whole fits into that. Our conversation centers around mostly T-cell receptors, and obviously I'll give you guys a background on the immune system briefly, but just as a heads up, this conversation was recorded very early, and I'm very appreciative of Martina for being able to make time to be able to converse with me about this fundamentally important part of the human body. Her schedule at this time of the year is such that she is extremely busy, and this was the only time that we could find. So the immune system generally, and we do not make much assumptions. We assume a basic understanding in our conversation of general immunological concepts. So students in undergraduate science degrees or medical degrees should be able to follow our conversation quite easily. But in case for those who do not have a scientific background that may want to be able to follow our conversation as well, I'll just quickly offer a background on the concepts that we touch on. So the immune system traditionally has been classified into two wings, the innate and the adaptive immune system. I'm just going to offer a simplistic definition of both of these two fields here. The innate system is the inbuilt system. It is fundamentally consistent of inbuilt structures that are ingrained into the body's design. And I know this is also true of the adaptive system, but for simplicity's sake, we'll just assume that. This consists of barriers, so physical barriers, chemical barriers, physical barriers such as the skin and epithelial surfaces that line the various visceral organs internally, and then chemical barriers like one's own tears, saliva, etc., stomach acid, etc. The adaptive system, on the other hand, traditionally has been believed to have two defining features. So these include memory and these include specificity. Now, there is obviously a caveat to that. There's some nuances involved, but for simplicity's sake, let's assume that specificity is governed by various functions as well as memory, and we don't really get into the finer details of this, but they do get mentioned. There is different cells that are involved within the innate and the adaptive systems. So the innate system cells consist mostly of cells that are of the myeloid progenitor lineage. These include cells that usually do functions such as phagocytosis, so that is engulfing, let's say, germs for simplicity's sake again. They engulf the germs and then they present these to cells of the adaptive system, which often are of the lymphocytic lineage. So without further ado, Martina Bonomi. Welcome to this special episode of the Den-versations podcast. I am joined today by a friend who has very kindly agreed to help me out with this, Martina Bonomi. Thank you very much, Martina, for helping with this.
Thank you for inviting me.
A bit about Martina. She, unlike the rest of us, did the right thing and majored in physics for her undergraduate studies at the University of Trento. Then she did a master's in applied physics with a major in medical physics at the University of Bologna. She then moved to Zurich to the Paul Scherrer Institute and did another master's thesis in proton therapy.
Yep.
She is now applying her fundamental understanding of the universe to biology at the Luciani Labs in UNSW. Thank you very much for joining me, Martina.
Thank you.
We're going to be talking today about T-cell receptor and some fragments of her work.
Mm-hmm.
So, Martina, let's lay the foundations for our conversation before we go in too deep. Start with the definition for a T-cell. classically, obviously, and I want you to add some layers to my definition, but classically it's been defined as a lymphoid cell that is able to recognize antigens in a MHC restricted manner.
Yeah, correct.
Could you add more detail to that? Because that's very imprecise. It's not the only...
Yes. So basically, T-cells are a part of the immune system that can develop. It's part of the adaptive immunity branch, and it's able to recognize antigens in a very precise way, and by doing that is also able to develop memory. So if you can get infected another time, your immune system can build a faster immune response and is able to clear the virus or pathogens or anything in much faster way compared to the innate immune system. And to do that, it has, like, T-cells have T-cell receptors that are actually proteins. They are formed by two chains that merge together when they develop in the thymus. And these receptors are highly diverse. And thanks to this huge diversity, that they can recognize any sort of antigen presented by antigen-presenting cells on the MHC molecule. So basically, T-cells recognize antigens only when they're presented by MHC molecules, and they do not actually recognize the whole antigen per se, but just a small fragment of it, that it's the peptide that for certain type of T-cells, so for example, there are two types of T-cells. There are CD8 positive T-cells. They are called like that because they express the CD8 molecule on the surface, and these characteristic, these cells are cytotoxic, so they're able to kill other cells. While there are also CD4 positive T-cells, which express the CD4 molecule on the surface. And they instead are called helper T-cells because they help other immune cells to perform effector function and regulate all the interactions and killing of things in the cell, in the body. So
generally, the definition of a T-cell then is a cell that has the T-cell receptor on its surface.
Exactly.
It's a lymphoid cell.
Exactly.
Develops in the thymus.
Develops in the thymus and recognize antigens
and has
with T-cell receptors.
And has effector functions as well?
Yes, of course.
Or can.
Yes.
Yeah. Okay. You've mentioned the T-cell receptor. I want to ask to build up on that as well. You said also it's a heterodimer.
Yes.
Disulfide linked.
Yes.
It has variable and constant regions, domains. Let's talk about the variable domains because they contain some complementarity determining regions.
Exactly.
Could you tell us a bit about these regions?
Okay, so as I said, T-cell receptors are very diverse because they are not born like that. When a T-cell develops in the thymus, it undergoes the VDJ recombination process, which basically consists of merging together different genes to make the variable part. So we have the T-cell receptor chain, alpha or beta, is composed by a variable and a constant domain, and the variable is made up of the joining of 2 or 3 genes. For the alpha chain, we have the joining of the variable and joining gene, so the V and the J together. While for the beta gene, we have the joining of the variable, the D domain, I don't remember the name, and the J gene, diversity, and then J gene. So, for example, for the T-cell receptor variable alpha gene, there exists 50 or 60 different genes. For the J gene, it's about 60, and for the beta variable is also 60, 70.
Wow.
And for the J gene is actually only 10, 13. So it's not that diverse, but putting together, you can create a lot of different combinations. But the most interesting thing is that when you join the variable and the joining parts together, nucleotides are randomly added or deleted at this region. So this creates another diversity part.
Yeah, so the length becomes more diverse.
Exactly. So the length is more diverse, and also the amino acid composition is very diverse.
Yeah.
And it's estimated that your own body can generates up to 10 to the 18 different T-cell receptors, but only 10 to the 12 are actually functional, because when the T-cells develops, the T-cell receptors must form and fold, and some of these genes are actually not functional. They're called pseudogenes. So if they are selected, maybe they don't create a functional T-cell receptor, so they are deleted.
Right.
And during the process of T-cell development, T-cells, before exiting the thymus and go to peripheral tissues, they have to pass a sort of quality check that is called negative and positive selection. So for first, there is the positive selection, where the T-cell receptor that is fully formed has to try to recognize peptides loaded on MHC molecules in the thymus. So if the T-cell receptors has enough affinity to bind and recognize that, they are selected. Otherwise, if it's not able to bind, they are deleted because they cannot do anything.
Yeah.
And during this is the first step. And then you go to the negative selection phase, where your body tries to delete the T cells that binds with too high affinity to antigens. Otherwise, if they go in the periphery, they might attack your own body and can cause autoimmune diseases.
Yeah.
So this is a double process where you can check whether T cells bind, but not too much.
Yeah.
So they have to, it's a process of self-tolerance.
Yeah. And I mean, we'll come to that concept in a bit.
Yes.
But so what's obvious here is that the T cells develop in a sort of multi-step process, right?
Yeah.
One step leads to the other, leads to the other, leads to
Exactly.
the other. And if the VDJ recombination does not occur, for example, the T cells cannot mature further, right?
Exactly.
And that's actually interesting, because a lot of biology students will probably know that we can actually apply this to creating mouse models that we can then use to study immunocompromised disease states. So, for example, RAG1 mice are mice that lack the RAG protein, which is important in the VDJ recombination.
It's the one responsible for stitching together all the different genes.
Yeah, it generates the nick. And then, yes, exactly. And when we knock out RAG proteins, you then get a TB/NK plus phenotype of mice that you can use to study the innate system, viral infections and transplantation, etc. Could you tell us, okay, what components of the CDR are important for MHC restriction?
Okay, so
because there's three per chain.
Exactly, there are 3 CD3s. There is CDR1, 2, and 3.
Yeah.
The CDR1 and 2 are diverse, but they are encoded in the variable genes. So they're diverse, but as diverse as the different V genes that exist. So if a T-cell receptor has 2 T-cell receptors have the same variable gene, they have the same CDR1 and 2. But for the CDR3 region, this is the most diverse because it's not encoded by any gene. It's just
it's the result of
random
junctional
process.
diversity.
Exactly.
Yes.
And the CDR3 region is actually the one that is thought to be responsible for peptide recognition, while the CDR1 and 2 are more responsible for stabilizing the contact of the T-cell receptor with the MHC. So CDR1 and 2, this is all knowledge that we know from crystal structures of T-cell receptors that have been generated. And the CDR1 and 2 basically mainly contacts on the alpha helices of the MHC, because I don't know if people know, but MHC has a sort of, it's a binding cleft sort of with alpha helices on 2 sides, and in the middle it sits the peptide. So the T-cell receptors comes on top, tries to recognize it, and does that with the CDR3 region to recognize the peptide, and the CDR1 and 2, it's like on the helices on the side
MHC,
of the MHC. Exactly.
Could you tell us a bit about, before we come into the TCR peptide MHC complex, could you tell us about MHC a little bit?
Yeah. So MHC molecules actually are the most polyformic molecules of the genome. For example, in the human genome, there has been sequenced and identified more than 6,000 different alleles. There are 6 classes mainly. There are actually more classes of MHC molecules. There is actually MHC class 1 and MHC class 2. So MHC class 1 is recognized by CD8 T cells, and MHC class 2 is recognized by CD4 T cells. For MHC class 1, we have MHC, actually for humans, we have HLA-A, HLA-B, HLA-C, but there also exists HLA-E, that is recognized by some innate like T cells like NKT or gamma delta or other stuff. And for MHC class 2 in the human, we have HLA, DQ, DR, and DB. So yeah.
Yeah, and the CDR1 and 2 then dictate a sort of binding to the MHC alpha helices.
Exactly.
What sort of angle do they bind at?
Okay, so T-cell receptors, when they dock on top of the peptide MHC, they do that with an angle that usually ranges between 40 and 60 degrees. And usually the TCR alpha chain sits on top of the alpha 2 helix, which is, if you look at the peptide from the N terminus to the C terminus, the alpha 2 helix is the one at the bottom, and the beta chain sits on top of the alpha 1 helix, that is the one on the top. So it's a bit dock on a certain angulation, so it's able to better fit and recognize the entire antigen complex. Yes.
Right. And let's talk about the CDR3 peptide recognition system then. As you've already alluded towards this already, but it's not precise.
Exactly.
There's just too many possible antigens that can be presented by the MHC complex.
Yes.
Than there are T-cell receptors. Right. And the T-cell then responds to this by a phenomenon called
cross-reactivity.
Cross-reactivity. Yes. Talk to us a bit about this then. And also, what are the potential implications for this, and how does the body deal with this? Maybe talk about.
I think that the main implication of that is, since when you have positive and negative selection in the thymus, of course, in the thymus, your thymus cannot present all the possible antigens that can be presented in your own body. So cross-reactivity might come into place in this case as well, because it tries to recognize something. If it's binding not too much, but also it has to bind, it can go in the periphery and recognize other antigens, or either the same antigens or also other antigens. So I think the first step is during the development in the thymus. So once a T cell passes this quality check and goes outside, is able to go around, patrol everything, and sensitize every antigens. And
the opposite of that would be that if obviously T cells were recognizing
just one antigen, it will be very, very hard to identify the right T cell receptor for the right antigen, because there are too many possibilities.
How many do you reckon there would be? Billions?
Yeah, sure, billions. I said 10 to the 12 T cell receptors. I don't know about peptide MHCs, because, for example, CD8 T cells recognize peptides on MHC class 1, and MHC class 1 presents peptides that are only 10 or 11 amino acids long, but MHC class 2 presents peptides that can be up to 20 amino acids, so it's very variable length. And you have this binding clef, considering like holding the peptide that has parts of it that goes outside. So we don't really know. T-cell receptor still recognizes the central part, but they can also recognize other
Around.
parts. Yes.
And let's talk about the specificity of T cells then, and then this will bring us back to the CDR3. They can recognize, they can tell apart two antigens that are one peptide apart, different from each other.
Yes.
How are they able to do this?
So actually, this is part of evolution because, for example, viruses try to escape from T-cell receptor recognition, so they mutate. And in some cases, T-cell receptors cannot recognize peptides with just one mutation, but in other cases they can. It's I think it's just about the 3D structure arrangement of amino acids in the T-cell receptors, that if you have certain properties of amino acids, so that make, for example, some more hydrophobic or hydrophilic regions or more electrostatic, it's just a matter of forces, I think, from a physical point of view.
Yeah. And talking of physical points of view, let's talk about biophysics of T-cell receptor to peptide pMHC binding. It's geared towards more so a specificity a target more than an affinity target, right?
Yeah.
So the T-cell doesn't, and could you talk to us as well about binding times here? The T-cell doesn't try to hold on to the pMHC for a long time.
Exactly.
Doesn't try to form any strong binding relations.
Yes.
So it binds and off. Could you talk to us about this? And maybe even introduce some slip bonds and catch bonds. Introduce us to that terminology.
Yeah. Okay. So when T-cell receptors recognize the antigen, they usually bind with either high or low affinity for not too long time, because compared to antibodies, for example, antibodies can stick to antigens for minutes or even hours and wait for other cells to come and kill the pathogen. But T-cell receptors, let's say, are a little bit more picky. They have things to do. So they just bind for a few seconds. And because the thing is that when T-cell receptor recognizes the antigen, it actually sort of rearranges inside the cell. So the T-cell receptor is always in complex with the CD3 molecule. So when the TCR recognizes the antigen, the CD3 rearranges in such a way that it can start phosphorylation inside the cell, and this phosphorylation starts a reaction that is able to activate the cell and differentiate, proliferate, and perform effector function and do things. So it doesn't require too long to do that, just the right amount of time. So usually T-cell receptors are found to bind the order of few seconds, but my lab actually a few years ago identified a T-cell receptor that is able to bind to more than 15 minutes to the antigen on average. And this is a T-cell receptor that recognizes an HCV peptide presented on HLA-B0702. That is an allele that is not that much frequent in the population. For example, there are some alleles that are more frequent in the population, like HLA-A0201. So we know a lot about this HLA, what peptides it presents, and also T-cell receptor that might recognize it. For the HLA-B0702 is not that studied and characterized. So it was very interesting, but the most interesting thing is that T-cell receptors that recognize these antigens were found in 2 people that were infected with HCV, but then cleared the virus. So, and the more interesting thing is that one of these 2 persons, the T-cell receptor clonal response to these antigens was made by just one T-cell receptor. So they just found one T-cell receptor with very high affinity that highly expanded and was able to clear the virus, this peptide. While in the other person, T-cell receptors recognizing the same peptide, the response was polyclonal. So different T-cell receptors recognizing the same peptide with the same high affinity.
And the same binding length.
Exactly.
Done. Right, okay.
So some people usually also talk and discuss whether T-cell receptor monoclonal or polyclonal response are more effective for clearing the virus, but this is an example that it can be both cases. So yeah.
How old were these people, if I may ask? I don't know if you know that.
Are in their 20s.
In their 20s?
Yes.
Right. And Caucasian?
I'm not sure, because this was a project where they enrolled prisoners.
Yeah. Okay.
And yes, so
Were they male or female?
One was a male and one was a female.
Okay, interesting.
Yes.
Oh, and do you know which one was which, by any chance? Sorry, I'm asking. I think
the female was the one with the monoclonal response.
Right. Yeah. Okay. Very interesting. Very interesting. So, I mean, once you've determined that there is this exception, then what benefits could that give us?
Also, in this case, is very debated. For example, we still don't know whether having a high affinity response or a low affinity response is better. There have been some studies saying that actually we don't really know what's the role of affinity in T-cell function.
Yes.
There have been some studies where they show that, for example, in cytomegalovirus virus, so CMV, people tends to mount a fast response. So the first response is made by T cells that have high affinity towards the virus, while low affinity T cells, they still recognize the virus, but they develop memory. So the T cells that last weeks or years after the infection, they are low affinity T cells.
Right.
So this could be, for example, a mechanism that, for example, high affinity are the first T cells that recognize the antigens because they need to do it fast and clear the virus, while low affinity maybe T cells maybe are outcompeted by high affinity, so they just stay there, they're activated a bit, and then they stay there and develop memory. So if you have another infection, they can mount a response, but they already know what to do, so they don't really need high affinity, they can do it low affinity, but this is just one case. So it's still very debated what's the role of affinity in T cells functions.
Yeah, I mean, it's interesting. I want to bring this back to whether our classification of T cells is outdated. Almost every year there's a new finding that is just turning our worlds upside down.
Yeah.
In your opinion, do you think that T cell classification is something that needs to be redone again?
If you mean classification as in subtypes of T cells, yes, because, for example, until a few years ago, T cells were identified mainly by flow cytometry in like a lot of like in a bulk way, by recognizing just proteins on the surface. But with the development of single-cell RNA sequencing technologies, now we are able to sequence the entire transcriptome of T cells at a single-cell resolution. That's what also my job is about. And when you do that, you can classify and group T cells based on the transcriptome and the genes that they express. And when you do this, actually, there is no black and white classification. It's not either this and that, but you can see T cells as a sort of continuum things because, for example, of course, there are, for example, CD8 T cells, CD4 T cells, and CD4 could be TH1 type, TH2 type, TH17, Tregs. But thanks to single-cell RNA sequences, we were able to identify, it's actually very hard to identify T cells that are just this or that, because maybe they can express some variable genes at different levels that can maybe look a little bit different, maybe in one population, but also in the other. So this could be also, you can identify like a differentiation path. So,
and is there any importance there? Is there significance to classification of T-cells? Does it help us in any way in our applications?
Yes, because if you can identify, for example, in diseases, some subtypes of cells that behave as they shouldn't, you are able to target them for treatments or cure, for example, autoimmune diseases as well.
Yeah. And I mean, one another interesting thing is our conversation, I should have probably prefaced, we're mostly talking about alpha beta T cells.
Yeah, exactly.
There's other types of T cells as well. So there is delta gamma.
Gamma delta T cells.
There is the mucosal associated.
Mate cells.
Yes.
And NK T cells that are T cells that they have an NK like phenotype. So they express receptor of NK cells and they recognize actually lipids on the surface of antigen presenting cells, so they do not recognize like classical MHC molecules.
Yeah.
They recognize lipids on other cells.
Yeah. And when we were talking about TCR pMHC binding, affinity and specificity, I was going to come back to whether this had anything to do with thymic selection, whether the fact that low affinity and low bind times were needed so that you could prevent the deletion of most self pMHC molecules.
Yeah, actually, the thymus is not a perfect machine, so it might still like leak some T cells outside.
Yeah,
but
and that would be dangerous, right?
Yes, it would be very dangerous, but I think that's cross-reactivity also comes in here because maybe if a T cell receptor recognize an antigen in the thymus with low affinity, with cross-reactivity you can recognize also other antigens, and you don't know whether for other antigens the affinity is still high or low. Yeah. So it's a bit tricky also in this case.
Yeah. Let's talk about crystallography. Yeah. It has helped us discern the T cell receptor structure.
Yes.
But it's now being replaced by cryo-EM. So could you introduce us a bit to crystallography as a technique?
Yes.
What it's been able to do in terms of T cell receptor biology and major histocompatibility, as well as its limitations. We'll go in a series.
Yes. Okay, so crystallography, as the word says, you need to create a crystal where you put inside your folded T cell receptors in complex with the peptide-MHC.
Yeah.
It looks simple, but it's not, because T cell receptors and peptide-MHC are not soluble proteins. So you have to make sure that you have to create them in a soluble way, because usually they are anchored on the surface of cells. You have to fold them in a soluble way, put together in such a way that they are able to bind and then make the crystal.
Yeah.
And to do that, you also need a synchrotron, because to get the structure in a crystal, you need a synchrotron with X-rays. And it's very time-consuming and very expensive method, actually.
Yeah. It's almost impossible to do. It's one of those things that is good for smaller proteins.
Yes,
right?
It's actually it takes years to do it.
With the T cell pMHC.
Yes, it takes a couple of years, 3 years, and it's very expensive because you have to express the T cell receptors and the peptide-MHC first on cell lines or something. And
also
then,
the T cell receptor pMHC is asymmetric.
Yes.
Yeah.
Exactly.
So it doesn't lend itself very well to the formation of crystals.
Exactly. Yeah. So it's quite difficult to do that. While cryo-EM is still not that easy, but I think it should take less time, but not that much. And basically, instead of making a crystal, you actually, I think you have to freeze your protein structure, your protein inside that in liquid nitrogen, and then with a microscope you take a lot of different shots of the structure. Of your structure.
Yeah.
And you can rebuild it, like putting together all the
And do you think there's a lot of promise there for the cryo-EM over crystallography?
Yes.
Yeah?
It's easier.
It's easier as well.
Yes.
It's cheaper, is it?
I don't know actually about the course about that.
Okay. And this brings us then to your current work.
Yeah.
It's very exciting stuff, cutting edge stuff.
Yes. Imagine getting a structure in just half an hour.
That is what you promised.
Yes. That's what I'm doing.
Yeah. Talk to us about AlphaFold, because you have worked with AlphaFold. A lot of people have heard about AlphaFold because AlphaFold has been in the news the last 1 year. Mm-hmm. Tell us why it's been in the news. What is it? Yeah.
So AlphaFold is a deep learning algorithm that is able to predict the structure of proteins. And it was very well talked about because the developers of AlphaFold were actually awarded Nobel Prize in Chemistry in 2024, so last year. Mm-hmm. And
when did they develop AlphaFold? Do you know?
The first AlphaFold was developed during the CASP competition in 2018, I think. But the cutting-edge AlphaFold version was AlphaFold 2 in 2022.
Right.
And then last year, AlphaFold 3 came out that actually improved the predictions with more accuracy, and also it was able to predict structures of complexes of proteins, because AlphaFold 2 was mainly designed for single chains proteins.
Yeah. If I can jump in. So I wonder if I can get your opinion on this. Usually, and I was talking to Professor Fisk last week, and he was mentioning to me that usually Nobel Prizes are given for something that has happened and has already proven its application over a period of time, right? What do you think? I mean, one of the backlashes that happened with AlphaFold is that people thought it was awarded too quickly. What do you think about that?
I think that a lot of people do not realize that AI has been in our research for a lot of time, because machine learning is AI, deep learning is AI, and I don't want to get me wrong with that, but also linear regression, some people call it machine learning, because you give parameters as input and gives you an output. So it depends.
Right.
But I think that's like predicting the structure of protein folding. It's something that people try to solve and wanted to do it for a lot of years. I think this first started in the 90s. And the fact that now with AI and deep learning, we were able to obtain a tool that can predict structure of proteins with such a high accuracy. I don't think that's too fast, because people try to do it for a long time, but with the new technologies, we were able to do it pretty fast, but it's not something completely new. It's something that people work on it for a lot of time.
Okay. So it's definitely commendable.
Yes.
Yes. You use it right now. But before we get into your uses of it, let me try to get the story behind how you started working with AlphaFold.
Yes.
When was this decision made that you thought this was a good time to start working with AlphaFold? How did that come about? Was it opportunistic?
I started my PhD last year, and the first 2 weeks when I joined my group, when I started my PhD, AlphaFold 3 came out. And everyone was like, oh my God, AlphaFold 3 is out. We should read the paper. And I read the paper and I was like, oh, that's cool. I want to try this.
Yeah.
And I still didn't know that my biggest passion would have been T-cell receptors, because I started with single-cell RNA sequencing analysis on CAR T-cells. And I was mainly looking at T-cells from a transcription point of view, so the genes that they express, how they differentiate, what is their role in disease, in cancer. And with single-cell RNA sequences, you can also analyze the T-cell receptor sequences. So just at the transcriptomic level, you can see with single-cell T-cell receptor sequencing, you can, for each single-cell sequence, the V, the D, and the J genes and the CD3 region, and you can do analysis of that. And this was also done for the T-cell receptors from the HCV patients.
Yeah. So somebody in your lab had already.
Yes,
Okay.
they already had sequences, and they already knew that those T-cell receptors were binding with high low affinity. And we were both me and my supervisor were wondering, oh, what's the difference between why do those T-cell receptors bind in such different ways? It would be interesting to see the structure. So, okay, let's try.
Yeah, and that's how you got into working with T-cell receptor
exactly.
structures. Right.
My passion for T-cell receptors started from the transcriptome, and then I moved into the protein folding because transcriptomics and sequencing is cool. But I think that a lot of people that try to, for example, there are a lot of machine learning algorithms try to predict the specificity of T-cell receptors. So what antigens they recognize, but they do that only with the CD3 sequence. But sequence, in my opinion, is not enough. The T-cell receptor is a three-dimensional structure, and binds in certain conditions, and it has some chemical properties, physical properties that make it recognize the antigen. So it's not just a CD3 sequence that is able to predict specificity.
So this is the physics coming out of you.
Yes. This is my physics anima coming outside.
This is why you do physics in undergraduate.
Yes.
Before you move into doing biology.
I actually started, I did physics because I wanted to do astronomy.
Yeah.
But I moved into medical field.
You moved into the right field. Okay. Tell us then, let's apply AlphaFold to the T-cell receptor.
Yeah.
Tell us about the usefulness of it, what you've been able to do with it, some cool features that you think you've been able to find with AlphaFold.
So AlphaFold, first of all, has been trained on the protein data bank.
Okay, yeah.
So to predict structure, it has to be trained on existing protein structures. And currently,
Yeah, and if I can jump in.
Yeah.
That's why it's important that we spoke about the limitations of crystallography, right? Because it then means that limitations that will come a bit later can be contextualized.
Yes.
Yeah.
So on the protein data bank, there exists only about 300 structure of T-cell receptors in complex with their antigen. That is definitely too few compared to all the possible TCRs that can be generated and antigens that can be presented and recognized. So I think the main breakthrough that AlphaFold can give us is that now we are able to predict as many structures as we want to better understand the basis of antigen recognition of T-cell receptors. For example, it took me quite some time, computational time, and quite some GPU time as well. And I'm sorry from UNSW, but I was ranked in the top 10 people using most of the GPUs from university this year.
So this is where 90% of the spending is going to.
Exactly. It's just for me. The billions of dollars of bills is just because of me. I'm sorry. I was able to predict structure for more than 1,000 T-cell receptor recognizing antigen.
Wow.
Yes.
Okay.
In just 2 weeks.
And are there any insights that you gained from that?
Yes.
Yeah.
For example, I'm analyzing now. I have folded T-cell receptor repertoires, recognizing different peptides. But for each peptide and antigen, I wanted to fold a lot of T-cell receptors that recognize the same peptide to understand if there is something common between all these T-cell receptors recognizing the same antigen.
Yeah.
And so I have 6 repertoires. I'm analyzing at the moment just 2 of them. And I actually found that there are common features for T-cell receptor to recognize the peptide. So it's a matter of final structure. I cannot just say amino acid sequence because there is some, for example, common motifs in the CD3 sequence or in the VDJ usage.
Yeah.
But it's not just that. I think it's more about the chemical properties of residues and the final structure that this complex makes. Because when analyzing some structures, I found that there are some cavities with some properties that actually should make make the binding true or not.
All right.
So
Interesting.
I think it's a little bit deeper than people used to do before.
So we're now actually gaining more.
But of course, these are just predictions. So there is no real structure for that, even though I was able to develop, like, modify AlphaFold in such a way that it can give me very high quality structures. So I don't know if you know, but for example, when you predict a structure with AlphaFold, it gives you a score for each atom based on whether the model is confident or not in the position in the 3D space of each atom and the relative position between each residue in the structure. And I was able to obtain accuracy for the structure for more than 90%.
90%?
Yes,
Wow,
that's
impressive.
pretty much.
What is the average confidence level that it gives you?
It depends, because for very, I don't want to say static, but compact structures, the accuracy is much higher compared to proteins that are more flexible or has some loops. Because, for example, when you try to fold a protein that is very flexible, has loops, AlphaFold gives you very bad scores.
Right.
But this can mean either that AlphaFold doesn't know how to fold it because it's never seen it before, or it doesn't have a proper structure because it's flexible. So the position in the space is just random. And for those proteins, for example, for flexible loops, you can use this also as a measure of flexibility of the molecule, not just it's bad.
So it's never a bad thing, huh?
It depends, because
Let's you mentioned loops. Let's talk then about the CDR3 loops, and let's talk about flexibility. How does that affect your predictions of TCR peptide-MHC complexes with AlphaFold?
So crystal structures actually have shown that when a T-cell receptors bind the peptide, the CDR3 and like the interface of T-cell receptors slightly changes to accommodate the binding. It's not rigid static to otherwise will just suck. So I'm not sure about that, because we don't really know what AlphaFold tries, like AlphaFold is a black box, so it doesn't really know what's going on. So when I get a prediction, I suppose that the final prediction that I get and the relative position between the CDR3 and the peptide is the most optimal one for the binding and the recognition. So I think that it tries to force, not force the binding, but tries to adapt the binding in such a way that it's what happens in nature as well.
Right. Yeah. So you mentioned just then that AlphaFold is a black box. If we talk about AlphaFold's limitations, do you think that these are actually limitations that can be overcome? Do you think they're just bumps along the road? Or do you think because of the fact that our crystallography data is incomplete and maybe our cryo-EM data is going to be incomplete for a while, we are going to really struggle to be able to predict TCR, pMHC models, for example? Deep
learning data, they learn, but they also need things to learn from. So
good response.
So I think I think actually people still generate crystal structure with crystallography or cryo-EM, because for example, papers, if you want to publish in Nature and you predict structures, usually the reviewers ask you for a real structure, so you use it as a validation thing. So if you have a T-cell receptors of interest, just one, you can still get a crystal structure. And the thing that actually people keep doing it, even with AlphaFold, to get real structure. And if you keep doing it and uploading on project data bank, AlphaFold should perform better. It should be retrained though. Because
how many updates of AlphaFold do we get in a year? So is it consistently automatically updating itself with new data?
I don't
or
think so. I think it's like the version of AlphaFold
that comes.
They are the updated ones, because AlphaFold 3 was trained as a cutoff date on the oldest structure that were deposited on the protein data bank up to 2021.
Yeah.
And I think all the others were not used for the training, but could be used for the testing for the model. And as we increase the number of structure on protein data bank, when AlphaFold 4 will come out, I think it will be even more precise, because it will keep into consideration also the other structure that have been added.
Yeah.
And hopefully maybe AlphaFold 4 might have a slightly different architecture that gives us better predictions as well.
Right.
Because the main difference between AlphaFold 2 and 3 is that they changed the architecture of the model, and it was able to perform much better than AlphaFold 2. So, yeah.
Okay. Almost done. Let's go into a more freestyle conversation. Okay. Thank you very much for that presentation on your work. It was really insightful.
I always love talking about T-cells.
Yeah, and here's another one for you then. Give us your best T-cell fun fact.
I don't know about fun facts.
Yeah,
I think it's
an interesting fact about the T-cell.
I think that the most interesting thing is that our body is capable of generating so many different T-cell receptors that we cannot even think of how different they are. But it still works. It's in my opinion, it's a complete mess, because it's just random combinations that actually work and are able to protect you from cancer, from infections, from everything.
It's very interesting as well. And it's like, it's not just the differences of the T cells as well. It's like the kinetics as well.
Yes.
So by not binding or by not creating a lock key model, by just having this weak binding, quick on and off.
I see it like Legos or a puzzle that you have to find the correct pieces that match together.
Yeah.
And
you're consistently going on and off, on and off, on
exactly.
and off.
Imagine having a puzzle with billions of possibilities. You try to make the puzzle and you don't know which one, but at the end you are able to find the pieces that go together. Yeah. It's sort of like that.
It's very mind-boggling, isn't it?
Yes.
Okay, what excites you then the most about your line of work? And you're not allowed to say AlphaFold 4.
No, what I'm able to do and discover with AlphaFold 3 is something that people just dreamed of in the past few years. They didn't even think it was possible. I'm analyzing thousands of T-cell receptors at a time. It's not easy. It's very difficult, because now I'm in the process of looking at each structure one by one, to see if I try to understand what's at the basis of this high affinity of this T-cell receptor recognition. And I think it's actually at the atomic level that makes it even more difficult. So it's very interesting in this way as well. Yeah.
So you're very interested. I mean, you're very excited that you're able to do things that people could only dream about back in the day.
I think this is the next generation of science and immunology. I've heard a lot of people saying, oh, AI will take your job and it will never go anywhere in medicine. Look at this. You can do a lot of things. I think the thing with AI is that you have to learn how to use it, so it will never take your job, because you have to be able to use AI for what you want to do.
Yeah, exactly. And could you tell me then, what are some applications of your work?
Like my main work now is to try to better understand antigen recognition from T-cell receptors. And if we are able to understand better this mechanism, we can, for example, engineer T-cell receptors that are specific for some antigens that can cause diseases, for example, cancer, but even autoimmune diseases. Because T-cells are good for killing things, like CD8 T-cells are good for killing things.
Yeah.
And in some cases, when it's those things is cancer, it's good. But if those things is your own body, it's not good.
Yeah.
So if you also are able to identify why those T cells recognize your own body, you can, or which one, you are able maybe to take them out and kill those cells that like to prevent autoimmunity.
And it's actually, I mean, it's of importance this because CAR T-cell therapy seems to have had quite a big success with beta cell malignancies.
Exactly.
We still struggle with it in terms of solid tumors.
Yes.
We still struggle with it in terms of other blood malignancies because there's such a thing as antigenic escape.
Yeah, exactly.
Right. And then we also have autoimmunity, which is a big other issue.
I think T cells, like, it's all about balance and tolerance, and there is like a balance between killing not enough, so you get cancer, and killing too much, so you get autoimmunity. So they have to be in the middle of these two things. And
that's what your job is.
Yes.
You're the person in charge of that balance.
Keep the T cells in the right balance, so you're fine.
Yeah. Martina, thank you very much for joining me.
Thank you.
It's been such a pleasure talking to you.
Yeah, I love talking about this.
Yeah, please do come back and we'll talk about T cells again.
Yes.
Thank you.